we need to collect plasma and others parts of this plasma for a very important questions and very critical questions in the hospital.
Welcome to the Evolving Past Alzheimer's podcast, where we are focused on bringing you information to help prevent from developing and improve from suffering with brain diseases like Alzheimer's and other dementias. I'm Dr. Nate Bergman, a physician and Chief Scientific Wellness Officer at Kemper Cognitive Wellness, and I'll be your guide on these sound waves. So whether you're a baby boomer who's worried that your brain wiring just isn't working like it once was, or you have a loved one with the disease, or you yourself have already been diagnosed and are wondering, what do I do now? You'll want to listen to this podcast. Each episode, we introduce you to the top doers and thinkers that are revisioning Alzheimer's, dementia, and just generally things in life's second half. If you have questions or comments, check us out on social media. To support us, go to patreon.com/evolvingpast and consider hitting the button and becoming a patron of this show if you find these episodes valuable. That's Patreon, Patreon, patreon.com/evolvingpast. So here we go. Let's get better. Welcome back to the show. We have a kind of an interesting and unusual format this week. So we're gonna talk about something called plasma exchange, and you're gonna hear a lot of fancy terms. And but this is research that's been going on for a while. But there was some research and results recalled to my attention by our co-host and co-producer for this episode. This is Dr. Brett Graham, who I have known. I've known Brett since he was a medical student at Lerner College at Cleveland Clinic, and now he is a second-year neurology resident down in Vanderbilt University in Nashville, Tennessee. Brett, thanks so much for being part of this today.
Yeah, happy to be here.
And thanks for taking the time to put together some of the research and prepare for this interview. Okay, so we're going to discuss the results of something called the AMBAR Trial, or the Alzheimer's Management by Albumin Replacement. And I'm just wondering if you can kind of give us a little bit of a kind of a nutshell of what this study is about.
Sure. So this was a pretty large randomized controlled trial, a phase 3 trial, so like kind of that last step that you normally give for approval of a drug or procedure, multi-center, occurring in Spain and the US, based out of Spain. And basically they took about 350 patients with probable mild to moderate Alzheimer's disease who were already being treated with standard of care medications, and randomized them to receive either plasma exchange or a sham procedure that mimicked this.
A sham procedure, I think you cut out there, so a sham procedure.
Yeah, a sham procedure that mimics this.
Sort of like a placebo.
Yeah, a placebo arm. And the plasma exchange group, they subdivided it further into 3 different arms with different doses of albumin and IVIg, which I think we'll explain in a minute what those are. So there's a total of 4 groups. And basically they went for a 6-week period. They had weekly plasma exchange procedures.
So this is like an IV going in. This is either going in their arm or in their neck, somewhere in their wrist or their arm, elbow, or in their neck somewhere.
Correct. So the majority of them, it was a peripheral IV, but some of them did need like a central line going in their neck. And then after that 6-week period, then for 12 more months, they had monthly sessions that were a little bit shorter than the previous ones. And so the 2 primary outcomes they looked at were the ADCS, ADL, and ADAS COG. These are basically kind of scores of quality of life, daily living, and then also cognitive scores. They measured a few other things as well.
So the outcomes are the things around which, the things that are measured around which the results depend, right? The outcomes are good or bad or somewhere in between. And, that's, of course, that's always a big question of, what were the basics, as you understand the basics of the outcomes of this study? Because we're talking about people between 55 and 85 who probably had Alzheimer's, or we call it a probable Alzheimer's diagnosis, right?
Right.
And there were, there's a there's many people in the, I mean, it was, what did they have? 347 people in the group that we
Yeah.
saw across almost 41 different places. So,
Yep.
not an insignificant number of people. I mean, not a huge number, but not an insignificant number of people. So, kind of what are kind of the baseline? What is the basics of the results without getting too
Yeah.
complicated too quickly?
Yeah. So the results were overall positive. The score that looked at the ability of people to perform their activities of daily living, they showed a 52% decrease in the decline over the study period in the treatment arm.
Okay, so let's talk about that activities of daily living, because that's a term that comes up a lot for you and I. If you're practicing medicine, this sort of ADL, um, activities of daily living can be broken down quite a bit. But I mean, I remember once I had Peter Holt. I had a mentor named Peter Holt, geriatrician in Kansas City, one of the teachers. He would say just, like, it's basically the things that allow you to get through the day, right? Can you get dressed on your own? Can you feed yourself if food is put in? Can you go to the bathroom and bathe on your own? So the basic things. And then there's sort of, I guess, they call them instrumental, or the finer things. Dr. Holt used to call them. What are some of the example of sort of instrumental activities of daily living? What are some of the other things that were measured here?
Yeah, so those would be things like being able to manage your own finances, being able to do things like work or do activities that require a little bit more higher level thinking and processing than just eating and drinking.
Okay, but these are basically the activities that define someone's ability. That we define, at least from a medical standpoint, right or wrong, as someone's ability to live independently on their own, so to speak. So if
Yeah,
someone has a loss of ADLs of some kind of activity of daily living, plus they have a brain or a cognitive problem or a memory problem, so typically we say they have dementia, right?
correct. Yeah.
Okay. So what did so going back to this sort of ADL function, what did this study show? Because that's a real
Yeah.
big deal, right? The ADL is a
Yeah.
big deal.
Yeah. Ultimately, this, you could argue, this is kind of really what we're aiming to treat, is people's ability to function and do the things they normally do. So it showed a 52% reduction in the decline in the treatment arm compared to the placebo arm.
Okay, so that's there's the rate of reduction. So there's going to be. We'll get into the fancy part of it. That sounds pretty good. 52% sounds pretty good. It sounds like a good chunk,
Yeah.
and we'll get into what
And
that
that
really
was,
means a little bit.
that was statistically significant as well. So that's something to, remember, important to know that it was unlikely due to chance. So
Okay, because that's the reason we do a randomized control. Just we'll try to. I know everybody's interested a little bit more in how studies, how the sausage is made, so to speak, over the last couple of years, especially in the Alzheimer's community, because we had the aducanumab trials, we had the adjuvant trials, and of course, like you know, Brett, we did a couple of episodes on that. And then we had Marwan Sabat on, who was so a leader at the Cleveland Clinic, and is really has been really pro that sort of family of medications quite a bit. And so like and then there's been some controversial approvals by our in the United States, at least the FDA, for some of these medications. So it's important to talk about statistics and methodology and from an evidence-based medicine standpoint, it really is important. And one of the first places usually people will go into reading a paper is not sort of after they look at kind of the abstract or the results. What was the conclusion in 2 sentences? Most people, I would say, maybe like you and I, will kind of jump down to the methods. Right? We'll jump down to the methods and say, okay, well, what exactly are we talking about? What was the methodology here? Because that's where a lot of the you can get hype in there. You can get a lot of misinformation and a lot of misunderstanding on how the study was done. Okay, so but I think importantly, let's take it back up to the 30,000-foot view here for a second before our guest comes on. Can you kind of tell us a little bit about, what was the intervention here? What was the thing that they were injecting? And it sounds like there were a few different groups of injections.
Yeah. And I also just want to point out that the other co-primary outcome, the cognitive score, they also showed a reduction in the decline, but it wasn't statistically significant. So I'm sure we'll talk more about that later.
So
And
say that again then. So because I'd
so
interrupted you a little bit there. So
yeah, so
the second big outcome that they looked at, the primary outcome, was a cognitive one, was one
yeah,
related to thinking and different kind of brain functions.
right. And they did show decline. They did show a reduction in the decline in that as well in the treatment arm, but it was not statistically significant. And
Okay.
so I think that might be why, overall the trial, why this is not like an approved therapy. It's they did not meet both of their primary outcomes
Okay,
technically.
so
So
reduction of decline. So that sounds
yeah,
like scientific doublespeak to me, right? I don't know. What is a
yeah.
reduction? So showed less
Basically,
reduction of decline.
slowing the decline is another way of saying that.
Okay.
So didn't
So that was one
completely
of the
halt it,
things.
but
Yeah, now that was one of the things that the Aduhelm or the aducanumab trials have shown, and some of the other drug trials have shown, that they say, well, someone who was already developed, already had a problem. Again, we're talking about people who all had um, the criteria for Alzheimer's disease, at least, without having someone pass away and doing an autopsy on them. They met the clinical criteria, because there isn't, as you know, any one test to say someone's got Alzheimer's disease, but they did meet an international predefined criteria. And then those folks were brought into the trial, and so participants are supposed to just get worse, right? And so these people got less worse, or the decline that they experienced was less steep, right? Is that what we're
correct,
saying?
correct.
Okay,
Yeah.
wonderful. All right, and perfectly timed here. We are just joined by our very special guest. This is Dr. Mercé Boada.
Hello, how are you?
We're
It's
excellent.
a pleasure to
Excellent.
be here. I don't know if you hear me in a very good performance.
We hear
It's
you.
okay?
We hear you perfectly, and I can see you
Perfect.
in HD. Thank you for making the
Oh,
time.
okay, fantastic. It
Now
was
we
really a pleasure to be here and to
uh,
talk with this very stunning topic.
So this is. I just want to give you. I want to introduce you, Dr. Boada. So Professor Merce Boada is a distinguished professor and neurologist at the University of Barcelona, and
Mhm,
founder and director of the ACE Alzheimer's Center in Barcelona.
mhm.
She's a chief medical officer. She's led over 135 clinical trials related to both Alzheimer's prevention as well as treatment. She's published hundreds of peer-reviewed medical journals, dozens of chapter authors. So she's a very, very big name in Alzheimer's, and certainly the topic of plasma exchange that we're going to talk about and kind of focus on today. But certainly her research and credentials extend far beyond that. Welcome to the show, and thank you again for making the time, Dr. Boada.
Thank you. It's a pleasure for me to be here and to talk about this topic. And thank you very much for your kind and introduction about my job or my vocation, because my vocation is Alzheimer's disease. Okay.
Excellent. So I have with me, who I've already introduced to our audience, Dr. Brett Graham. He is a second year neurology resident down at Vanderbilt University in Nashville, Tennessee here. So he'll be kind of assisting and kind of asking the questions here. I do want to give you a minute or a few minutes just to kind of frame your experience in the world of Alzheimer's and Alzheimer's research, and particularly what led you to think about plasma exchange, both albumin and IVIg, which we'll talk about. What kind of led you in your thinking to doing the AMBAR trials?
Well, that is a beautiful and long history, because we are start working and with plasma change and albumin replacement nearly in 2004. In 2004, the first approach in this new approach in to treat on to understand to treat Alzheimer's disease patients was focused in two key points or in two pillars. The first pillar is the wide experience from the Grifols company about the donors, the blood donors, because for to obtain plasma and to obtain blood in the blood donor centers, they use the plasmapheresis.
Okay, so let me just, I
And
want to make sure our listeners are tracking with us. If I understood you, so Grifols is a company that does plasma. They get the plasma from
mhm,
donors, from people
donors,
with
okay,
donor, right? So they
is
get,
is
and what is plasma? Just
what is plasma? Plasma is the recipient in our body that it's composed by water, liquid, and proteins, and is used to mobilize and to circulate a lot of products about around our body, and they have a lot of the implications in our health, and to keep our body in the very good fit, because they transport nutrients, proteins, and elements of the blood, like red blood, plaquettes, and white blood, and another is to immunoglobulins, the globulins, human globulins. It's
yeah,
transported, it's moving around these. Sometimes when I talk with the kids in the schools,
yeah,
is the big river. Is the big rivers like Amazon, like Mississippi. That is our circulation, and
the
it's
big rivers.
full of our the nutrients. One of the important things, in order that you understand how it's Grifols and the importance of this company around the world, is because we need to collect plasma and others parts of this plasma for a very important questions and the critical questions in the hospitals, or we have in one an catastrophic situation like a war or like a tsunami or like a volcano, because we need plasma to recover patients.
Yeah,
And Grifols is one company. It's not a pharmaceutical company in terms that you can understand the what is the requirements of Grifols around the world, because it's one hemotherapy company. They working with hemoderivates, producing hemoderivates for the hospitals, basically for the hospitals. And the first pillar to arrive into AMBAR is Grifols have a good experience and very extensive experience, because they collect tons and tons and tons of plasma around the world
yeah,
in order to supply this gold component for our health.
yeah.
Eh?
It's a global
And
company. It's a huge company.
it's a huge company. And one of the really the knowledge in this company is how is the role of albumin in our body. And one of the roles of the albumin in our body is to transport. It's to transport components, is to transport components. One, the characteristic of this albumin is because it's the most abundant protein in our body, in the plasma. The second question, but it was the very well known use of the albumin, is because they maintains the intravascular volume. When I lose volume, I need to replace with liquid, to solve one critical situation. And one of the most uh, abundant and most frequent in order to recover volume and to keep and maintain intravascular volume is albumin. The other is because it's a main transporter in this Amazon, on the Mississippi, on the Nile, and they transport fatty acids. Is one of their missions. Another is a, a lot of drugs. One of them is like when I use dopamine for the Parkinson disease, because it's linked with the dopamine. Another is metabolins, metals is transported and add to the albumin. And the other question is albumin is the major transport of the A beta protein, a beta amyloid. Another question is later, with the time, with investigations, we know that albumin can be affected by the one oxidation process, one glycation process, and to translate parts of the sites in this protein and have one a place, a role in the immunoinflammatory effects. For all the reasons, Grifols and the team of research think about that albumin could be one approach with plasmapheresis, one approach in order to treat Alzheimer disease patients.
Okay, so this is
Eh?
amazing. So this is really interesting. So what we were, my colleague and I here, Brett, we're we're talking about. Okay, so what was actually done? So with some of the patients in the trial that we're going to talk about, they received in
Mhm,
exchange of a blood product called plasma, which you explained beautifully. Thank you. An albumin, as I understand it from you here. So it may play a role in binding out amyloid from the
mhm,
brain, which is
mhm,
thought to be a major contributor, or in
mhm.
some cases, just a signature of the developing
Alzheimer disease.
disease
Okay.
of Alzheimer's, and maybe as an antioxidant in terms of responding as a sort of to this immune and inflammation, sort of an immune response and inflammation that's often characterized in Alzheimer's disease as well. So that's
As well.
the first intervention as far as the plasma exchange or the blood exchange that people were getting was albumin. And then there's this a second component you're going to talk about too, it sounds like.
Okay, the proof and concept, the first approach was critical to know that if patients with Alzheimer's disease, patients with dementia, severe dementia, advanced dementia, moderate, in this case is moderate, can support, can accept this type of treatment. It was a big challenge. Because just at that moment we are starting to work with monoclonal antibody using the pumps, no? And that is a big question that could be this kind of the patients possible not tolerate 1 hour or 1 hour and a half uh, under this treatment, that it's necessary to remove plasma remove plasma
Right.
with one peripheral access and replace plasma. That it was the first challenge, this first challenge with 10 patients was really successful. The second question here, in order to follow up and understand the history of AMBAR, is we follow these patients for 1 year, following these patients for 1 year, and I'm sorry, one moment,
Oh, it's okay.
and I'm sorry, one moment, and we um, we can find that these 10 patients keeping, and it was increasing the cognitive performance, and we replicate and make one extension of the first proof and concept project study, and this proof and concept study that it was published in the drugs in the drugs um, paper, uh, offer the Grifols in order to have data, solid data, considering that the solid data is and proof of concept for the
Right.
pharmaceutical company to follow up, and really past this pilot extension, the first confirmatory clinical trial was the phase 2. And in this phase 2, we include not so much patients, 45, and these 45 patients was shared with US, two sites in US and two sites in Spain, and to include another strategy in terms of frequency and volume of tool replacement. And we use one strategy. He was 2 times a week with a total plasma change. Total plasma change means that I can clean or remove more or less 2 liters per person, 2 liters or 2 liters and a half or 3 liters. It depends the body mass, and replace this one with albumin in the concentration of 5%. The first period, 2 times per week. Second period, 1 time per week. And in terms of maintenance, in order to find what is the best dose for the treatment, it was one time every 2 weeks.
1 time every 2 weeks for the albumin?
No, I'm sorry. 1 time every 2 weeks. Okay. And in the total of the pilot, the phase 2 study was six and 18 treatments, 18 periods, 18 treatments. And the results of this one, it's very well tolerated for the patients. No important side effects, very well tolerated side effects, minor side effects, and very well tolerated. And the second, we test that the improvement in cognition in some domains. In cognition, the best one is language, and the best one is executive functions, and the performance in the daily activity was confirmatory to the proof and concept. And
Improvements in
finally,
language and executive function and some of the
and
retaining of daily activities.
okay, and daily activities, because daily activity, it was confirmed and contrast with the exploration for the patients. That it's the formal way to test the daily living activities, and with the caregivers. And both are considered that the patient feels better, and the family understand that improving, because the most important for the people who are working in Alzheimer's disease is how we can offer to the patients, and how is the demand for the family and for the patient, and they will feel better. They want to get autonomy, to keep their autonomy as long as possible.
Yeah.
And that is published in the Alzheimer disease, uh, journal.
Mhm.
And I remember that it's in the 19, I don't remember very, very well. Maybe
That
it's
was so.
in
In other words, you've got this.
1990.
You've been out
Okay.
for this. You've been at this for a while, since
Yeah,
2019.
it's
Yeah.
okay. And we describe the methodology, the plan, and the demography, because our patients, it was not my cognitive impairment, and it was very, very mild Alzheimer's disease. It's people with underdiagnosis of the Alzheimer's disease. And I want to here clarify one crucial point. All the patients that be involved in AMBAR project, and previously, the diagnosis is under the clinical criteria and the phenotype.
Yeah.
The phenotype. We don't use the biomarkers to include these patients. Another thing is biomarkers was acquired during the protocol, but not using to involve patient. It was under the clinical criteria, in order that if AMBAR was successful, it could be the most global treatment, because in every one site or in every one country, if you have a good clinician, a good neurologist with a team of the neuropsychology, we we can to offer this treatment without no biomarkers. And AMBAR,
And I want to make
and
just a
sorry,
one clarification. No, it's just I want to make sure everybody's tracking with
yeah,
us because
thank you.
one time when we talk with scientists, especially if you've been doing science for a long time, I just we try to convey the results to people that are smart, but the lay public. So, you, Dr. Boada's sort of moving us through how we set up for this current publication within sort of the AMBAR stages of trials that we're talking about,
Mm.
um, and and I think feasibility and reproducibility, tolerability. And so she's talking about, well, if we've proven that in smaller groups this was tolerable, it's reasonably safe. And then, instead of using what Dr. Graham and Dr. Boada and I had talked about before this, which was like a biomarker. Sometimes those need, we need to do a lumbar puncture or an MRI or sometimes a PET, certain kinds of expensive PET scans that may or may not be available everywhere. And it sounds like the reason that you used a clinical diagnosis, meaning a physician and maybe a neuropsychologist, as the criteria for receiving this treatment was because it seemed that would if the results were positive, it would allow more people access to this as a treatment, and as opposed to someone who had who needed biomarkers done that may or may not be accessible to many folks still.
That is a good resume, summarize very well the intention and this new approach. This new approach, because new this approach is that at the end, one treatment can be affordable for the systems, for the health systems, because in Europe, it's even though maybe in North America and South America, in Europe they are the very incomes countries and the others no. But the medical doctors is a good medical doctors because we know how to do with our patients. And this approach is approach, even though the plasmapheresis is very well known around the world, even though and it's not cheap, it's not cheap. But it's affordable for the hospitals in every country.
Yeah.
It's one approach in order to include or the increase the benefits from every kind of the patients with this diagnosis. Mostly of these patients are in the moderate state, and that is really too easy to check these patients in our memory clinic in other hospitals,
Yeah.
because it refers for the symptoms, signs, and symptomatology during the time. That is a good question, in order that this approach is easy to understand, and affordable to administrate and to use the therapy.
Dr.
Well,
Botta, can I ask a
yes.
question? So you used it. You also, in addition to exchanging um,
Mm,
the way you described the plasma exchange, this blood
mm.
product, and then giving people albumin, you also in the trial we're talking about, there was a group of people. There were, of the four groups, the sham group, the people that got only the lower dose of albumin. There was another group that got lower dose of albumin and another product called IVIg, intravenous
Yes.
immunoglobulins. Um,
Okay.
can you talk about that and why you included
Yes.
that as well?
Okay, very, very, very smart question. Because if you look the protocol that it's in our publications, the first period that is we name intensive period is when we use one total plasma change every week during 6 weeks without no immunoglobulins, because we follow the previous experience. But in the AMBAR protocol, we introduce one new terminology, but it's a new frequency of the administration of the processes and the albumin in terms of the maintenance period. And in the maintenance period, you use the low volume plasma change. And the low volume plasma change is more or less a half volume from the previous one. But here, because it's the more long time, we're thinking about that we clean proteins that I mentioned it when we introduced the the um, the the concept and the hypothesis of the AMBAR. And with these proteins, they are immunoglobulins. And in order to give the most safety protocol is include immunoglobulins just in case we decline or we clean the volume or the levels of the immunoglobulins during this period and induce uh, immunoglobulopenia or induce the infection diseases.
So
And,
you were
and
worried, if I can just clarify, so you were worried a little bit that if you would, we know that the immune systems of folks with Alzheimer's disease are somewhat impaired. That's not controversial. That's well described. Exactly how to exactly what, and we don't know so much. We're learning, but like just so that you didn't
Mhm.
over clean the immune system and sort of remove some of the important elements, you gave them back immunoglobulins, so proteins that are very involved in the immune system. You gave that to them after those initial 6 treatments with just the albumin plasma exchange. They were
Yes.
given either a lower dose, a higher dose, or some didn't receive it at all of these IV or intravenous immunoglobulins.
Okay, and that is one concept that we want to include in order that offer safety, a safety protocol, and the company was talking about this one with FDA, and the FDA recommend to in this design that use only one of the arms without immunoglobulins to follow our experience in the previous one. And really in our experience, because in Fundación ACE, in our center, we collect all the patients from Catalonia,
Okay.
for the procedures, and we reach 1,500, nearly 500 plasmapheresis. And when if you read the paper, the people he was under the immunoglobulin replacement supply, eh, and supply, they have less infections. But by the contrast, the people under the immunoglobulins increase the side effect like dizziness, uncomfortability, because that is very well known when when you treat with immunoglobulins. And that is an important point in order that when you want to talk about the real world evidence that we are working, how we are managed to move and make a translational from the clinical trial to the real world evidence. And the group only with albumin without IVIg, they increase a little the infections normally, but reduce the adverse events uh, led by the immunoglobulins.
So they had so the group with that got the albumin had slightly less side effects or significantly less side effects,
Less
but also had maybe they had a little bit less immune system
immunity.
support conferred
Okay,
to them. Okay.
okay, okay. And all this experience lead us to understand better how to do in the real world.
Okay.
To manage this one, and solve this important point. As you know, we are used the 3 different arms in the maintenance period. That is a long period using a high dose of albumin and immunoglobulin. Really is 20 grams of AGV, or less immunoglobulin, 20, we replace with 20 grams of albumin with 10 grams of IgV. And finally, the clean arm without immunoglobulins is using the low dose of albumin in order to understand how is the effect of the albumin, how is the effect of the removed plasma, and how is the effect the immunoglobulins in the middle in this clinical trial. Because AMBAR is focused in the two strategies that is working all together. For one hand, is to clean to the toxics, metabolins, elements, proteins in our body that is used in the critical room, even with the COVID, to clean the storming of the cytokine storming because we clean. And the other is the effect to add a new albumin and to preserve and continue this albumin have their functionals properties during our life because one of the properties is the albumin is in the elderly people and even in the Alzheimer people, albumin is under one process of oxidation and glycation. And if I remove plasma with albumin and replace with pure or young albumin without no trace of the A-beta amyloid, I restore the functionality process of the albumina, like I use young albumin to rejuvenate this plasma in order that keeping these effects.
Fascinating. So I think we're tracking with you. I want to allow for Dr. Graham to ask some questions. I think he had some kind of pointed questions around the trial results, because it's exciting, and it does feel like that they're it's IVs and the kind of the field of neurology, and certainly in Alzheimer's and maybe in other dementias as well, but Alzheimer's seems to be a little bit ahead, is going to be sort of this kind of an immunotherapy, chemotherapy kind of approach. At least is what we can access at most major hospital systems once there really is good from data from some of these trials. Brett, can you want to kind of step in and
Yeah.
Hello.
So I want to ask you, hi, Dr. Boada. It's an honor to be here with you. I want to ask you, let's get into the results a little bit. And there's a lot of them. So I want to kind of preface this with kind of a summary or just like the parts that I think we could focus on at first. There were two
Oh,
co-primary
thanks.
outcomes that you looked at. So there's two things: the activities of daily living, which Dr. Bergman and I were talking about this before you came on, and then there was the cognitive scale, and on both of those, it showed about a 50% slowing in the decline in the treatment arm. Um, One of those, the activities of daily living, the p-value was just barely, it was past that significant level of 0.05, and the other one, it was just
Mistake.
barely
Okay.
missed it, 0.06. And then, there was a whole bunch of other subscales that you looked at, and pretty much all of those were positive, showing a beneficial effect in the treatment arm. Some of the ones that really, kind of impressed me also was, the global impression score. There was basically 100% less decline, so people in the treatment arm stayed the same. And then in one of the kind of subdivided groups, the mild group, one of the scores, the CDR, the Clinical Dementia Rating Scale, actually people actually improved in the mild Alzheimer's group. So those seem like a lot of positive results, but there was, because one of those was like one of the co-primary outcomes was technically not reaching the statistical significance level. I think maybe that's why, at least from my perspective and talking with other colleagues, this was perceived as overall a kind of equivocal or negative trial. So I want to kind of hear, how would you describe the results of the study, and what do you think about what you
Hi,
found?
Brett, and it's a pleasure for me also to be with you and to share these inputs and well, the your suggestion. You are very well defined the results of AMBAR. Okay, because we have the co-primary endpoints, and the co-primary endpoints, when we analyze all the subjects, like it's 322 from global, very mild and mild Alzheimer's disease and moderate Alzheimer's disease. In ADL, we reach really one p-value acceptable. I believe more than acceptable, with one effect size or one effects in the last decline, nearly more than 50%. That is not bad, because when we have the graphics in front of me, I always say, if we extend gave one extension in this program, maybe we can to reach more effects, because we have no huge number. If we compare with the maps, a huge number of the patients included in the AMBAR study, and the period is limited to the 14 months. But if we prolong to extend, maybe the difference, or we increase the number, is one of the strategies to find a good p-value. And when we talk about the other scope, that it's important, that is one co-primary endpoint, we don't reach the p-value, but the side effects reach more than 60%, nearly at 66%.
Which group gets the 66% side effect?
Yes, in the other scope is cognition. Is in the tests that we talk about the cognition. And of course, in one of our co-primary endpoints that don't reach to be positive. That is the discussion, and that is the it's not a fighting, but it's the discussion with academics. How it's here, because some groups they said that is a failure, because one of the key points it's don't reach a B positive, against that the others in terms of the we reach one side effect. Really, I don't want to use astonishing, because astonishing for me is when I talk about the CDR and clinical global exchange. But we are reaching really the effect. When we analyze by the different arm in terms of the phenotype, mild Alzheimer's disease and severe Alzheimer's disease, there are some difference that it's very difficult to understand. There are one not published paper, but we have this data, and we are presented these data in the other forums, when we compare US with Spain, because that is very interesting to understand how it's collecting the patients in US and how collecting the patients in Spain. They are a big difference. The difference is US collect more mild Alzheimer's disease, basically collect mild Alzheimer's disease. And in Spain, we collect moderate. Maybe because in our clinical memories, not in mine, but around Spain, the patients arrive a little more advanced. Okay, a little more advanced. And when we compare the different samples in terms of phenotype, but we compare the differences in ApoE and difference in the CSF-A, amyloid, the
Right.
difference is in Spain, our patients are ATN, or at least 8 positive, and US not, because it's a little more mild. We need to understand a little more this difference from the countries, in order that to understand a little more what is happening when we are working with mild Alzheimer and we working with severe Alzheimer. Of course, in ADL, the mild Alzheimer could be improved that it's improved a little in cognition, because cognition have a very correspondence with activities like the living and my capacity of the cognition. But when we are working with more moderate, cognition is less,
More
and the
advanced, more
effects
advanced
is more abundant, and it's less in the effects in the LDL. And I believe that this is one stone in our shoes, in terms to be very colloquial in the AMBAR, because I want to reach one p-value nearly to be statistic positive if I have more time. But in contrast, when we talk about the CDR Santa boxes, it's incredible, the p the p-value, because our p-value needs no doubt, and it's extremely positive, because the difference from one another, the placebo group and all patients, it's really it's the tens, it's not conversion at the end, it's completely diversion. Right? It's amplified at the end, and we have one effect site, the 70%, more or less, it's a 17%, and in the clinical global impression or change, that it have a value in all the clinical trials related to Alzheimer's disease, it's more impressive, because the effect site is 100%. We are improving or no modifying. Well,
So yeah, actually, Dr. Boada, you're making a few really good points that I
yeah,
wanna just
yeah,
summarize again too. So first of all, um, when we're doing a clinical trial, like uh, we have to choose
yes,
something to say that this is our primary
2 points.
endpoint. This is what we're primarily focused on, and some of that is just it's a decision, and sometimes you kind of, in a sense, choose wrong, or you realize, like, oh, there's much more impressive effects in all these other things, but we happen to choose
Yes,
these ones. So and that sometimes, unfortunately, that means those other um, side benefits kind of get discounted, even though they're very positive. The other thing you mentioned is the length of the p value. When we determine it's statistically significant, if we're looking at the curves, we're looking at kind of
mm,
how far are these
mm.
people separating, and is that likely due to
No,
chance
no,
or not.
you have,
And and so that's, yeah. And so like you said, the longer you look, the more those curves are going to separate, and the more people you have, then the tighter your estimates for each of those numbers in each group can be. And so this is very common, like in things like heart disease. You study people for 5 years or more to be able to see any difference, because the you know it, diseases like Alzheimer's, the change is gradual, it's slow, and so we're not going to see huge differences in the groups right away, especially if they're smaller. And so that's, I think, a very good point to point out, is like, yeah, if we had extended this trial longer or had more people in it, then things probably would have been even more, stark differences. Or, and then, uh, finally, there was this distinction you were talking about, U.S. versus Spain, and you were saying in Spain there's more moderate people in the study and more mild in the U.S. And actually, if I remember correctly in your paper, most of the benefits that and most of the major differences you were seeing was in the
moderate.
moderate subgroup of patients. And my interpretation of that was probably just because those people declined fast, more over the study time period. I don't know if you have any other suggestion
Yeah,
why we might not see as big an effect in the mild group compared to the moderate.
yeah, all use comments at extremely adequate and accurate into the discussion of this clinical trial, because they are always, we are working in the two hats, in the two hats. But one of the hats that we need to worry is to confirm and to confirm our hypothesis in positive or negative, following the rules of scientific and academic rules. And as you mentioned, of course, that is my hypothesis. If no, always is one. If we collect more patients, if we prolong the clinical trial, it's possible to find a good results in cognition. Okay, and that is a really one important discussion. But I want to ask Brett another and to change the in this party, to change some things in this party. And when we designed the plasma change study in the proof of concept, we consider following following that it's appear in the in the in the clinical trials around the world focusing Alzheimer's disease, that the two key points, the primary key point was ADAS-Cog and LDL. But when MAPS is really entry in the market of the clinical trials, as impose like the most brilliant and expectation about the treatments in Alzheimer's, they change for ADAS-Cog, for CDR sandboxes. If we change this arbitrarity, because that is our choice, and when one company takes a choice, take a risk that could be failure, and it's impossible to change this one. That's its one reality, and it is published, and it's we have to discuss. But if we have the opportunity, or could be the opportunity in the past to change at the scope for CDR, our clinical trial was positive. That is the risk when you design one clinical trial in the time, focus in the knowledge, focus on the regulatory agencies ask for the clinical trials. And if we need to design another clinical trial, focus in this clinic, in this approach, the plasmapheresis, or in the other approach, in order that we manage very well the clinical rating scale as a instrument, as a battery that offer a huge vision about the performance, clinical performance, functional performance, and cognitive performance of the one patient. Maybe if I decide a new clinical trial, I change the ADAS-Cog, because ADAS-Cog was designed for the inhibitors from anticholinesterase. It's 30 years ago. It's a 30, and our knowledge about Alzheimer's disease, it changed completely. We are introduced biomarkers. We know the risk factors. We know that the genetics, how genes can to increase the risk, and how genes can to protect this population. But one thing that is important when we are improve our knowledge in my cognitive impairment is to detect that we have um, um, a group of the patients who move very fast to decline and converse very fast, and they are the others that it's very, very stable, even though when we talk about the hippocampal sclerosis, that we have a good diagnosis, a good phenotype, but never change.
Yeah.
Now, if all this knowledge we are include in the new design of the clinical trials, I completely sure that my design now could be a little different. Could be a little different,
Yeah,
but
so I
that
think
is that. No, I have
you're
this. That is the results, and I have I have no other solution. We need to understand. We need to understand how we can offer to the patients.
Can I jump in, because I think you guys
Yes.
are Brett is zeroing in on a really important question, and a question I think a lot of people have. So like somebody like you, Dr. Boada, who has seen a lot of, I would call them hype cycles. Maybe the promise of something new, and then it didn't pan out to the next thing, and what you and Brett have been sort of going back and forth on are, okay, well, what tools do we use? Do we use the ADAS-COG, the ADAS-COG? Do we use a clinical dementia rating summary of boxes? So these things might be quite technical to most of our listeners who are, let's say, not physicians, not researchers, but it's been Alzheimer's field has been challenged for a long time as to how to collect. There's a couple of questions. How do we measure results that are meaningful? Are they just quality of life to care partners, to individuals suffering with dementia? Are they neurocognitive assessments? Is it some combination? And basically, Dr. Boada is saying, at some point you just got to call your shots based on the time, based on the regulatory environment at the time, and then we have to understand the results. And so sometimes it's not those primary outcomes that are the most interesting, even if they, whether they performed or they underperformed. But it's some of the sub-analysis and some of the little signals that are not always bright, big signals. So I just want to make sure we didn't lose any of our listeners, and I don't get out of the way, Brett, for you to ask some more questions.
Yeah, I think that's, and it sounds like, Dr. Boada, I mean, and I tend to agree with you. I think overall, if you look at the results overall of the AMBAR trial, it seems pretty promising, especially when you compare it to all of the previous trials of Alzheimer's drugs. I mean, the fact that we have in some markers complete stabilization, like no decline, and some we even have a small improvement. I mean, it seems like a big deal. I guess some additional questions I had is, like, do you think these effects are likely to be durable? So is this something that people would basically, they start and then for the rest of their life they're basically have to get a monthly plasma exchange, or do you think that and do you think that would continue to produce benefit long term, or at some point is it going to be kind of not
I believe
working as well?
yes.
What are your thoughts on that?
That is a very smart question, and a very understandable question for the group of the persons and the professionals who are working with Alzheimer's disease, because the question is, how is in the long-term treatment? No? That is how is happen if you offer me the possibility to treat during 40 weeks, 40 months, or 2 years or 3 years. No? That is a question, Brett, that always asking us to the families and the patients. Because and the answer is, when we start to treat with, or we are working, because I am older than you, and I was in, and that sometimes is one advantage, or another it's not, because you are more future than me. And when I was start to working in the clinical trials with donepezil and with ribastigmine, and really in the 24 weeks is accepted for the FDA, the results. six 6 months, 6 months the results, because we need to have something in our hands to offer the patient. Later, the second the second study that offer more understanding, that can offer the inhibitors, was the Nordic donepezil study, and the Nordic donepezil and prolonged during 1 year were in the treatment. And during 1 year, the effects, it's maintained. And the past 1 year, we treat the patients with donepezil, with ribastigmine, galantamine, even though with the nemantine, who appeared, it was launched in 2003, for 2, 3, or 4 years. What is the stopping rules in order to cancel our compromise with the treatment when the patient declines, and worsen and worsen. And we said, well, this drug is not working. Hmm? The question is the same from the new drugs that cortisone and the new drugs that the MAPS or anti-tau. The clinical trials I always describe like one corset. One corset is start and to stop and finish. And I need to live and to understand this data. But in the real world evidence, I can't to prolong this treatment forever. No. Oh, fantastic it was forever because patients never worsening. But there could be one time that you, Brett, and me, we said, well, this drug is not working. We need to finish this treatment to change from the other, to include another, and that is the state of art of the medical doctors. When we are working in today with the foot in the land, and every one of the patients is completely different, it's completely different. The follow-up of these patients is not so intensive that when we are working in the clinical trial, and we need to acquire experience, security, to trust in our actions, and to include patients in one treatment. If you talk to me about if in Spain or IMA approve Biogen, Dr. Abada, do you want to treat patients with Biogen? I try to select very well the patients, to understand very well the situation, the clinical situation, and the comorbidities of this patient. And of course, if that is my opportunity to offer some new and to some hope to this patient, in our institution, we want to treat patients. If you talk me, Dr. Abada, you are treating patients with Alzheimer's disease in the real world with plasma change? Of yes. Of yes. In our institution, now we are including in the treatment 67 patients. Treat in one the best adaptation in order to offer the most comfort to the patients and to understand how it's happened in the real world. The real world for us is when one patient said, oh, that is a Christmas period. I don't want to treat. In the clinical trial, that it's impossible. But in the real world, I can say, no, 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 no. In the no, it's well, I open my window because that is your choice. No, or sometimes because one
Yeah.
patient have one another problem, and we stop the treatment, take away, and include the new process in the next days, on the next week. That is the real world evidence offer us the possibility to understand for a long term one treatment. And if
Yeah.
we are honest with our knowledge, if we are honest in our possibilities to treat, and we are honest with our patients, when this treatment it's not working, this treatment must to be cancelled. That is one.
Right. So
Hmm.
presumably, at some point, based on the data we have, the benefits may no longer outweigh the expense or the risks or things like that, and we, as the clinician, can decide when that time might be with the patient themselves. And so, really, what's lacking is that long-term real-world data. I guess another
Mhm.
question I had, this is uh, a very I think in terms of side
Hmm.
effects, mostly it seemed pretty well tolerated. But I guess one of the big concerns that I was wondering about is this is a very expensive procedure, and especially if you add IVIg, which is often in shortage and costs a lot of money as well. And when you're talking about tens of millions of people with Alzheimer's worldwide, is it feasible that we would be able to, if this was approved today, would we be able to offer it to everyone? And is that just feasible from kind of a cost and a supply and demand standpoint?
Well, a key point. Interesting question, because this question is talking about the future. It's talking about the future. How to do? How to do? Well, in Spain now, in our country, that is not pay for our public health system. Plasmapheresis is pay for the other diseases in the system, and it's completely free, because our system, our public health system, is some different from the US system. All the citizens in Spain have the opportunity to be entry in the one liver transplant, cardiac transplant, completely free. The treatments from oncology are free, and the treatments and the process from the diagnostic in the Alzheimer disease is free, because it's paid for the state and for our taxes, right? And our taxes. But plasmapheresis is free at the hospitals for one critical situation in myasthenia, in the another neurological diseases, the high critical toxicity in the for one intoxication, external in the intoxication, but for the chronic disease like Crohn, and like hypercholesterolemia, familiar hypercholesterolemia malignant. That is completely free, and the hospitals pay for this one. But Alzheimer's disease is a novelty, because it's a new challenge for our system. What is my point of view, my interest, my hope, and my challenge in our institution as a leader of this beautiful project that part of my life, 40 years of my life, is devoted to understand plasmapheresis, is to increase little by little, step by step. I have not, I am in not a fast track. I am like a turtle, little by little, to understand the process, to increase my knowledge, the experience, to take note about how is thinking the patients, how is thinking the families, in order to put on the table this new approach from the payers, the health payers authority. Could be it takes 1 year, I don't know. Could be it takes 2 years, I don't know. But my end point, my key point, is that all the citizens in Spain do have this criteria can be included in this clinical trial, paid by our authorities at the total, pay 50%, pay another, I don't know how kind of this modality, like in cancer, but the citizens and the families and the patients with Alzheimer's disease can have one opportunity to increase their quality of life and their future without disease, or with a minimal disease. That is my challenge. But that is my crazy personal challenge, because I believe that one clinical trial, that we have not areas, we have not adverse events, because the first adverse events was focused in the catheter, for the catheter, because at the first we uses central catheter. Now always is peripheral,
Oh,
because
so you're
US is,
saying initially you were talking about adverse events. Initially, we were worried about the catheter
yes,
is in the
more,
central, the one that goes in the neck,
yeah,
and,
and
and now the
yes,
standard is the peripheral, more in
yeah,
the
but US have not problems, because they use peripheral, and in Spain you use central. Well, because the company wants to test what was the better. No? And really we have local reactions from the catheter, but in terms of hypotension, minimal. In terms of anxiety for the patients, nothing about syncopes or about presyncops and dyspnea, because the vagal symptoms is as a consequence that in one moment our body have less volume because I am removal, and they have 5 minutes that I need to replace. And all people, because all people, more of them, they are very sensitive in this less volume, and that is the presyncom. But I have here the data, and the presyncom from the total we have from the total plasma the procedures. We don't reach 30 patients with the presyncom. That is a minor effectiveness. One of my recommendation,
Just for everyone
yes,
on the, I just want to make sure what you're talking pre-syncopal. We're
yes,
talking about just that feeling of about to pass out, but not completely passing
okay,
out, and so kind of heart rate going up and feeling
feel bad, no?
maybe
Okay,
anxious and yeah, feeling
feeling
bad.
bad,
So you're saying that about maybe up to
yes,
30% maximum
no,
is
no,
that's
yes,
happening,
it's
and it's usually it's not a super
no,
serious
no, no,
long
no,
term thing. It's just
and yes,
uncomfortable.
it's in terms of percentage, they don't reach 1%. 1% is only 30 cases. 30 cases.
Oh, okay,
30,
30
30
cases. That's a
cases.
good clear. Okay, so only 1%.
It's
That's
much
much
different.
different. Okay.
And
Yes.
in the very expert hands, the most important is to build one very expert team, an expert team from the diagnosis that it's usual. That I believe that it's one, of course, for to choose and to describe very well the patient, the best patient for this treatment, and the other is to work with a very expert uh, apherist that managing very well the device, managing very well the access, the venous access, and the team of the medical doctors who supervise the process and take the actions if one adverse events appear during the treatment, and to understand the treatment. The understand the treatment is how the measure of safety I I have to be in my hands before to start the treatment. The all the parameters from hemostasis, because I use calcium and the fibrinoise is decreasing. I have to and to um, every time to have one AKG, because we monitorize the AKG in order that nothing appear during the process. Another thing is magnesium, calcium, and in our center, the immunoglobulins. In order that immunoglobulins previously is decreasing, stop, we delay, wait and see, in order non use the immunoglobulins to be more safety and most costly, no, less cost, no, affordable for the citizens. That is that little by little we can to uh, to acquire data and to present in one academic forum in order to describe the new situation and discuss with the audience and the experts how is in the real world this new approach to treat patients with Alzheimer's disease.
Good. So yeah, in other words, there's not like a there's definitely some monitoring that goes along with this to make sure it's safe overall. If it's well monitored and well
Okay.
managed, it can be safely done. But you do have to check levels of certain electrolytes and like magnesium, calcium, sodium. You have to also monitor levels of clotting
Okay.
proteins and things like that. And so,
Yes,
yeah,
and
that's a good point.
hematocrit, and hematocrit, because
Hematocrit.
hematocrit is the parameter that design how is the volume we can to change.
Yeah,
It
hematocrit.
depends to the hematocrit. It depends of the hemoglobin and the body mass. And every time, if every 2 weeks, it's really it's no change. It's not change because our previous data, hematocrit is stable, albumin is stable, the kidney function it's very stable. But when we have 1 month, 1 month for the other treatment, something could be happen during this time, and we need to recheck in order to offer the most safety safety conditions to the
Yeah.
patient. That the most important is that the patients feels that they are controlled during before the process, during the process, and the after the process.
So they say, and I know I want to be sensitive to time, and we're just past our time that we had allotted here. I do want to just kind of close out here, ask Brett if he's got any other pressing question, and I also want to just ask you um, if you think that the albumin, or even if this is just sort of like your intuition, or it's sort of an anecdotal observation, or it may not be true from what you've seen, but people that are generally living a healthier lifestyle, so eating healthy, controlling diabetes. You mentioned, albumin can get beat up pretty bad uh, through numerous reasons: high blood pressure, inflammatory cholesterols, problems with blood sugar, and that can kind of deregulate the body in other ways. Like when you think about people that have had, they're exercising, they're eating well, and they're participating, they're not smoking, maybe have good blood pressure control, kind of doing other things that we know are good for Alzheimer's disease, either in prevention or reduction of symptoms. Have you had any sort of anecdotal experience thinking, okay, people that are working really hard in other areas of their life and getting IV albumin plus or minus IVIg, do they seem to fare the same or any better than folks that are just coming in for the albumin, the plasma exchange, plus or minus the IVIg, and are still going home and with diabetes and no changes in diets and still smoking cigarettes, maybe, that's kind of thing, the lifestyle. It's a question I think a lot of our listeners are wondering, if there's a medicine or a pill that can just fix it. Everybody wants that. But my hypothesis, the working hypothesis, is that probably we'll be better with doing both. But I'm wondering, you're in the trenches, seeing people, year over year, decade over decade, and if you can make any comparisons to what you see anecdotally, even if you haven't studied it per se.
Well, that is an open question, is an open reflection. Is an open reflection because in the future, not my generation, but maybe the generation of the breath of my grandsons, could arrive in a good chip, in a good health situation to the 100 years, and that is our future. 100 years, and 100 years is focused in two things. One is style to life, and style to life is to reduce vascular, neurovascular risk factors because it's the same, to keep uh, uh, a good physical exercise in order to increase our metabolism, endorphins, establish a good diet with low fats. With low fat, that it's one important risk factor. No smoking, no drinking, blah 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 blah. But another thing in this sense, that is another, is to be alive in our social activities. For cognition, I need to establish a good relationship with the other. We are making now one excellent exercise of psychostimulation, because we ask it, you think one question, I need to answer this question, and we are very active in terms of intellectual or cognition. For me, it's cognition, but cognition also is emotions. COVID, COVID changed our emotions because we are living isolated. And the patients with Alzheimer's disease, it's worsening because they have not human contact. We are humans. I need to talk with Brett. I need to talk with you. I need to talk with my husband, with my grandchildren, explain about my books, my impressions. I need to love people.
Yeah.
And I need that people love me. That is one pillar. The other pillar that we are understand with plasmapheresis and with another groups like the group of Tony with Corey in Stanford, it's to know what is happened with the proteins in our body. And with the aging, our protein is changing. It changes. If we can restore it, these proteins, with plasma change, because plasma change is one treatment like one octopussy. They have a lot of key points. It's not only one. And if we can to restore it in the 50s, not in the 80s, in the 50s, we can to restore it, our liquid component, our proteins, in order to decrease oxidation, glycation, tal, maybe when we reach 100 years, we can reach these 100 years happy with a good, no, happiness is important, happy with a good life and with a good future.
Oh, I love
More
it.
than
Thank you.
10 years.
Well, beautiful. So I want to thank you, Dr. Boada. So, and Brett, of course, thank you for the preparation and the insightful questions and the wonderful discussion. So, in addition to albumin and healthy lifestyle, love, the Dr. Boada has prescribed love and happiness. As
Proper happiness. Thank you very much, Brett. It was a pleasure. I hope to meet you in one place in the very, very nearly future. Thank you very much to offer me these opportunities to talk about the albumin. Albumin is the white part of the eggs, and it is life. Okay? Okay, thank you. Thank you very much. It was a pleasure.
So that's our episode. I hope it was useful to you. Check out the show notes on iTunes or on our website where we've summarized the key points. If you find the information here valuable, please consider giving us a 5-star rating on iTunes and leaving comments in the comments section. It will really help us bring this message to as many people as we can. If you have questions or comments, connect with us on social media. Finally, to support us, go to patreon.com/evolvingpast and consider hitting the button and becoming a patron of this show. That's Patreon, Patreon, patreon.com/evolvingpast. Thanks, and talk to you next time.